क्या लिखा है? To impart a knowledge of general carbon and nitrogen metabolism in the plant in special reference to starch and cell wall polysaccharide and biological nitrogen fixation and assimilatory pathways. First is carbon metabolism. Another, in carbon metabolism, we will understand synthesis of sucrose. How sucrose is synthesized. Generally, in your fundamental of biochemistry or in introductory biochemistry, you have read only glucose plus fructose is equal to sucrose. Similarly, synthesis of sucrose, how much, where it is synthesized in plant, how it is regulated, what are the component is required. Then regulation of sucrose phosphate synthesis. Another chapter or another topics we will understand the transport of sucrose, phloem loading and unloading of sucrose in phloem. Okay. Then synthesis of starch in leaves and seeds. As you know that what is starch? Starch is the storage polysaccharide found in most of the cereals. And many plant species having this starch. Generally, starch is also synthesized in tuber crops too. So we will understand the synthesis of starch in the leaves and seed. Then we will understand concept of transitory starch. Another topic after that in carbon metabolism we will understand synthesis of fructose. Fructose is what? Tell me what is fructose? What is fructose? Fructose. Fructose is a monosaccharide. Ketose sugar. Am I right? Ketose sugar. C double bond O. So you have to remember the structure also. Okay. Every monosaccharide, every amino acid, every individual components you have to understand as well as to remember here, not here. Okay. So here is what is fructose. So I think here it is bind. Okay. So 6 carbon sugar with ketose. What is that? CHO in place of C double bond O. So synthesis of fructose, then synthesis of galactomannan, that is also sugar. Okay. Then synthesis of triose sugar, triose, tetrosis, pentoses. So monomer unit are joined together. Raffinose, series of raffinose, trehalose, that is also in our course. Another topics now. Up to this, you have to understand carbon metabolism. In carbon metabolism, fructose, oligosaccharide, and sucrose synthesis. This is much more important. That I will ask in the examination also. Then nitrogen cycle. Now nitrogen cycle, as you know that in environment also nitrogen cycle is there. In soil also nitrogen cycle is there. Okay. So both how the nitrogen is assimilated in the soil, how this nitrogen is utilized by the plant, where it is synthesized, what is the biochemistry of nitrate assimilation, and how it is regulated. What are the enzymes involved in nitrogen metabolism? We will understand. Then, which pathways? Glutamine synthetase and GOGAT and GDH pathway. These three pathways is much more important for GOGAT pathways is very important for examination point of view. Then two molecules or metabolites, urides and amides. I will also take in practical also how to determine urides, how to determine the amides as a nitrogen transport compound. Repeat this word.
Urides and amides as nitrogen transport compounds.
Repeat this word.
Uridates and amide and nitrogen
Urides.
uridates.
Urides and amides. as a nitrogen transport
transport
compounds. Then another word, chemoautotrophy in denitrifying bacteria. I will give you a what mean by chemoautotrophy. Okay. Then what mean by nitrification? What mean by denitrification? What are the bacteria are involved in denitrifying bacteria? That is also in our course. Now, majorly important biological nitrogen fixation is important topics for our course of this carbon and nitrogen metabolism 5, 10. Then what is the functions, how it look like, structure, then regulation of enzymes, nitrogenase, NIF genes and their regulation. What are the biochemical bases of legumes? Biochemical basis of legumes. Leguminous crops are rich in protein. Why this is rich in protein? Because of these nitrogen fixations at hetero. They having the root nodules. So leg hemoglobin is also found there. And that is in our course also. Rhizobium biosynthesis. Rhizobium symbiosis. Gene involved in synthesis of these legume. So these are the important glossaries or terms or meanings you have to remember. I will ask first question in every first 1 hour course, second 1 hour, practical also, and final also. Terminology is important. Okay. So we will start with the sucrose synthesis. So the definition is sucrose. What mean by phloem loading? What mean by transitory starch? What is galactomannan? You can add these what is with these words. Okay, that can also ask like this. Define nitrogen cycle, define Gogard pathways, draw the Gogard pathway, draw a nitrogen cycles, give the example of oligosaccharides. Such question can be asked in the exam. Now you tell me, urites and amides are involved in what? I have repeated 2 times. And each one of you have already read that sentence. Urites and amides, the compound which are involved in nitrogen transport. Okay, should I repeat? Urites and amide as a nitrogenous transport compound. Remember this word. Chemoautotrophy, denitrifying bacteria. Example of denitrifying bacteria can be also be asked in the examination. NIF gene symbiosis. Define symbiosis. What are the NIF genes? How the rhizobium biosynthesis or symbiosis, carbon metabolism. Okay. Now importance of this carbon metabolism in the plant and for the biological molecules. So you can read terrestrial photosynthesis. Terrestrial means what? Above the ground. Okay, you can see with your eyes. Terrestrial photosynthesis is responsible for the fixation of about 50 billion tons of carbon per year. How many?
10 billion, 50 billion tons of carbon per year.
50 billions.
50 billions.
Hmm? Million 10 lakhs. Billions, I'm asking about billions tons of carbon per year. How this carbon is synthesized? Who is synthesized carbon? Whatever you are taking or burning. Okay, plant is also synthesizing. And we people utilizing more resources and converting to a carbon. So majority of this carbon is deposited in the polysaccharide as rich soluble polysaccharide, mitigating anthropogenic increases in the atmospheric CO2 by sequestering this sequestering. Repeat this word.
Sequestered.
Sequestering carbon in plant tissues with long half life. So ultimately into it converts into what? Soil organic matter. Full form banao. SON, SOM, soil organic matter. Why in our literature they are getting SOMRAS is very important. They don't know what is English, but they know soil organic matter. Okay. So generally, photosynthetically fixed CO2 is ultimately source of carbon for the synthesis of what? Now you read this sentence
The
loudly.
majority of this carbon is photosynthetically fixed. CO2 is ultimate source of carbon for the synthesis of nucleotide sugars, and there are several potential routes for their production.
Now repeat this word without seeing that. Here. See here. Now tell me which are the reaction is involved
Fix
to fix
CO2.
CO2? Which reaction is involved? Photosynthetic. Photosynthetically fix CO2. Huh? That ultimately, that is ultimately source of carbon. Now, why this is essential in human body, in plant also or animal also? Because they are synthesizing two compounds,
Nucleotide sugars.
nucleotides and sugars. Nucleotide sugar is that ribose. Okay. So there are several potential route for the production of this. In illuminating source leaves, carbon fixed by photosynthetically in chloroplast and is exported
Into
cytosol as triose phosphate, glyceraldehyde 3 phosphate. Okay, so these leaves carbon fix. How carbon is fixed by this photosynthesis, where it is synthesized in chloroplast. Then it is exported. Exported means
Transport
transport into where? In cytosol. At what compound? As triose phosphate. In cytosol, there are condensed to make hexose phosphate, including glucose 1 phosphate, which is the substrate, glucose 1 phosphate. Hmm? In cytosol, they are condensed make make hexose phosphate. Hexose phosphate means glucose. Glucose is the hexose. Glucose 1 phosphate. So, which is the substrate? Substrate, as you understand in the enzyme. Enzyme plus substrate, ES complex, and plus you will find a product, E plus P. Okay. Similarly, here is also which is the substrate for the synthesis of UDP glucose. This UDP glucose is synthesized by UDP glucose pyrophosphorylase is the enzyme, and this enzyme name or enzyme classification number is given as a 2.7.79. Now, first topic, sucrose. As you know, sucrose is the most abundant disaccharide, major product of photosynthesis. Now its characteristic. It is a non-reducing sugars with limited chemical reactivity and thus and thus reason can be asked in the examination. Red one. Utilized as a transport and storage molecule in the most plant. Why? Sucrose work as a transport molecules or a storage molecules in most of the plant species. Why?
That's why it is called
non-reducing sugar.
and limited chemical activity.
Because this is the reason.
Limited chemical
Because it has a limited chemical reactivity. Why this limited reactivity of sucrose is that? Because it in their structure, when glucose and fructose both joining group are joined, so no free group are there. So it is called as sucrose is a non-reducing sugar. That can also be asked in the examination. Okay, reason. Now sucrose synthesis. As I have started with, that is predominant in the leaves. But ability to synthesize among all the plant cells. So it is non-reducing sugar, widely distributed, utilization among higher plant too. Trehalose. Trehalose, the only other non-reducing disaccharide that is found in nature and play a comparable role in insect and fungi. Whether it is found in plant or not, trehalose, yes, where in certain plant species it is attacked by fungi or insect, you will find a traces of trehalose. But trehalose is not as such synthesized in any plant species. Okay, so cedar wood oil or some scented oil, scented volatile oil is very important in certain species. Wood oil, I don't know the word. Okay, certain woody plant species having infected with fungi and it gives scent. Okay, so sucrose is not only a pivotal player in the assimilation or storage distribution of photoassimilate but also osmoprotectant. किसकी बात हो रही है? किसकी बात हो रही है?
Sucrose is
हाँ,
right.
sucrose is not only a pivotal player in the assimilation, storage, distribution of photoassimilate, but also act as a osmoprotectant that accumulated during periods of abiotic stress. Abiotic stress, which are the abiotic stress? Cold, drought, then osmotic drought. So, for example, osmotically stressed plant, this is given the example of spinach, accumulate sucrose apparently through the activation of SPS. Now, the new topics, that is, you read loudly.
Role of sucrose synthetic function and location in product. Sucrose is the end product of photosynthesis and the primary sugar transported in the phloem of most plants. Now, sucrose synthesis, SUSY is short form, is a glycosyl transferase enzyme that play a key role in sugar metabolism, primarily in zinc tissue.
Stop. Now, why? Which enzyme is responsible for sucrose metabolism or sugar metabolism? Primarily in the same tissues. Sucrose synthase. Sucrose synthase. Loudly speak two times. Sucrose synthase. Susi. Okay. Now the another question, that transport in the phloem, most of the plants, which molecules are found? Sucrose. Sucrose is the end product of photosynthesis and it is work as a transport molecule. Enzyme sucrose synthase is a glucoside transferase enzyme, another name. Okay, that play a key role in sugar metabolism, primarily in the sink tissue. Source to sink. Leaves is a source, and seed is a sink. You can say like this. Sucrose synthesis catalyzes the reversible cleavage of sucrose. Cleavage यानी तोड़ना, breakage. So sucrose catalyzes the reversible cleavage of sucrose into fructose and glucose and either uridine diphosphate glucose. This uridine diphosphate glucose, UDGP, UDPG, uridine diphosphate glucose, or ADPG, adenosine diphosphate glucose. So the end product of sucrose, cleavage by which enzyme? So you cannot write like sugar. Sucrose synthesis, or another says glucosyl transferase. Okay. Now glucosylate sucrose into a fructose, thereby fructose either uridine UDPG or either ADPG. तो आप रिएक्शन बनाओ, ये सुक्रोज सुक्रोज
Sucrose into
Sucrose is acted by sucrose synthase
into fructose.
and it changes into fructose plus uh, UDPG
very
or
good, or
ADPG.
okay, this is the sentence what you have written, right? So you can write like this also.
Enzyme
Huh?
sucrose synthesis.
You can write like this also. Pointly, these how the sucrose is synthesized. You can write this way or this way. Okay. The end product of these sushi are available for many metabolic pathways.
ATP production.
So as you know that it work as a energy production, ADPG. Okay. Then primary metabolic productions, synthesis of complex carbohydrates. So plant sushi activity or sucrose synthase has been shown to play a pivotal role in sucrose metabolism. Name itself indicate. Same words I am repeating. I am repeating source to see. Okay. And sucrose synthesis. Now all the enzymes are protein. So sucrose synthase is also a protein. Where it is found? It is found in cytosol or adjacent to plasma membrane. That can also be asked in the examination. What? Which question?
Where sucrose synthesis is
Sucrose
found.
synthesis protein are found.
Hmm. I can also ask like the which enzymatic protein are found in cytosol or adjacent to the plasma membrane. So sucrose synthase isoenzymes are found in cell wall, mitochondria, vacuoles और are bound to actin. अब बोलिए कहाँ कहाँ पाया जाता है? Where it is found?
Cell wall, mitochondria, vacuole or actin.
Bound.
लेकिन सुक्रोज सिंथेटेज iso isoform.
Isoform. Not
Not yes.
Isoform.
Isoform word is important if you are writing when you are writing with cell wall mitochondria vacuoles. Okay. But if it is asked where it is primarily.
only cytosol and adjacent to plasma.
Okay. Now you can understand what is the difference. Sucrose synthesis enzymes involved in several metabolic pathways already been taught to you. Now कौन सा ऐसा pathway? Which are the pathways involved by this metabolic? So first is for starch, for cellulose and cellulose synthesis. Now you make one question from this. Yes, you, you, you.
Sucrose synthesis.
Which are metabolic pathways? और ऐसा word use करके बोलो.
Which are metabolic pathway? Different.
आप यही हैं,
सारी तो है सर.
बोलिए.
Which metabolic pathway enzyme sucrose synthesis? એટલે મીન્સ કે કયા પાથવેમાં સુક્રોઝ સિન્થેસિસ થાય એવું જ કહેવા માંગે.
No, no, Keva nahi. I am asking that you have to speak a question, make a question from this sentence. Now you try.
Yeah. In which metabolic pathway plant sucrose synthesis?
What is the role of sucrose synthetase enzyme in metabolic pathways? Which are the metabolic pathways are involved or used by the sucrose synthetase enzymes? And what is the role of sucrose synthesis enzyme in which are the metabolic pathways? You can make your own sentence. Otherwise, you will not understand anything. How many times? Difference, sir. I'm reading three times, four times. I cannot understand what you are speaking. Okay, that can also possible. Therefore, first
Yes.
start, ask question to yourself, ask question to sentence. Okay, so to play a developmental and possible signaling roles in sync carbohydrate flux, vascular tissue, and merismatic functioning. Now I am asking, what is the role of sucrose synthesis enzyme? Where it is located? Answer me. First you.
Carbohydrates flux, vascular tissue, and meristem function.
First you have to write it. There are various metabolic pathways are involved. These are the metabolic pathways involved sucrose synthesis. Okay. Then you enlist this. It is also involved in plant development. It is also involved in it has a role in signaling roles. It has a role in zinc as a carbohydrate, and it is also found in the vascular tissues and merismatic functions. Where it is located, generally it is found in the cytosol or adjacent to the plasma membrane. But their isoform is also found in
Cell wall,
mitochondria, cell
mitochondria,
wall,
vacuoles.
vacuoles. Likewise, you have to answer. Otherwise, what will happen? Same as UG people. Okay. So this is the sucrose synthesis pathways in the plant. How major reaction that you know very well. UDP, this UDP, UDP glucose plus fructose 6 phosphate that will convert into a sucrose 6 phosphate plus UDP plus H plus. Now and sucrose synthase. So see, UDP glucose plus fructose, both reversible. Now reversible reaction. Sucrose plus UDP plus H plus. So sucrose synthesis can be catalyzed by two distinct enzymes in higher plant. Which are the two distinct enzymes? Now you tell me.
Sucrose phosphate synthase.
SPS and SUSI. What? Two enzymes, SPS and SUSI. Now tell me what is the full form of SPS?
Sucrose phosphate synthase.
Synthase, not
Synthase.
synthetase, synthase. Okay. SPS and SUSI. Sucrose synthase and sucrose phosphate synthase. These take place in the cytosol. Disaccharide, sucrose is a disaccharide of glucose and fructose that place in the cytosol of mesophil cells. Sucrose synthase activity in the developing xylem vessels or fiber may be particularly important for what? Cellulose synthase, cellulose synthase. Okay, so important for cellulose synthesis needed for the construction of thick secondary walls. As in starch synthesis, glucose residue is activated as a nucleoside diphosphate, UDPG, ADPG. Okay, although in this case, UDP glucopyrophosphorylase enzyme. So this is the pathways you can see in the picture also. Fructose plus UDPG, that's NDP sugars, hemicellulose, cellulose, and pectin is formed. Okay. Similarly, in the respiration also, exose phosphate is involved. Sucrose is a translocation too. So sucrose production by photosynthesis in the source as a leaves is here. Okay. So upon arrival in the syn tissues, sucrose can be unloaded to phloem sympathetically. This symplastically via plasmodesmata. Remember another word, symplastically via plasmodesmata or apoplastically, Simpo, simplastically, apoplastically. Apoplastically, sucrose is taken up either directly by sucrose symports, SU, okay, or after hydrolysis to glucose and fructose is by the cell wall invertase, okay, cell wall invertase with excess sugar and imported by a sucrose transport protein, STP. So intracellular sucrose can be metabolized by sucrose synthase, SUSI, okay, cytosolic vascular invertases. So sucrose directly produces UDPG, which is substrate for synthesis of cellulose, hemicellulose, pectins. Mostly, this is the precursor, nucleotide sugars precursor needed for hemicellulose and pectin synthesis. Glucose and fructose are the phosphorylated hexokinase, that is HXK, okay, and fructokinase, that is fructokinase, FK, respectively, resulting hexose phosphate and used UDPG, okay, or UGP. So this is all about the sucrose synthesis, sucrose catabolism, and synthesis of cell wall. You have to write like this. Okay. Now SPS. What is SPS?
Sucrose phosphate synthesis.
Okay.
Synthesis.
Synthase. Sucrose phosphate synthase.
Synthesis.
Another way,
Sucrose
Sucrose.
synthesis.
Sucrose and SPS. Okay. So sucrose is synthesized in the cytosol by consequent activities of two enzymes. One is SPS, another is SPP, sucrose phosphatase. Sucrose phosphatase or sucrose phosphatase synthesize, catalyze reversible transfer of glucoside unit UDPG and fructose 6-phosphate that already been taught to you. So in plant species that several regulatory phosphorylation site is available. So in both soluble and membrane bound proteins or membrane bound forms is responsible for catalyzing both the synthesis and degradation of sucrose. So this is all about sucrose degradation, sucrose synthesis, and in contrast in the chloroplastoma, this pyrophosphate is not present. So that whatever the given underlines that can also be asked in the fill in the blanks. Okay, UDP. UDPG pyrophosphorylase reaction is reversible. Which reaction is reversible? Whether it is invertase or this UDPG. Now SPS. That catalyzes transfer of glucose residue, that is UDPG, to the fructose 6 phosphate, and that will forms a sucrose 6 phosphate. Okay, so therefore overall reaction can is irreversible process. Whether it is in question mark, still it is not proven. Sucrose synthesis is not expected to play a role in the leaf starch synthesis. अभी तक मैंने स्टार्च के बारे में नहीं बोला, तो ये आपको याद रखना है. So this is all about the adenosine diphosphate liquid, ADPG is the main molecules convert into a starch by the starch synthesis in the plastic in chloroplast. The main starch synthesis pathway starts where
Chloroplast.
with two molecules of triose that is produced by the photosynthesis. One is fructose 1,6 biphosphate, and this fructose 1,6 biphosphate transported to fructose 6 phosphate by the fructose biphosphatase to fructose 6, which is then converted into a PGI. PGI means glucose 6 phosphate, that phosphoglucoisomerase. Phosphoglucoisomerase, PGI. Glucose 6 phosphate. Yes.